Hey, what are we gonna do for our project? Justin.
Justin. Justin. Justin.
Yeah? Yeah, what?
What are you thinking about, man?
I don't know. I was just thinking, like, how does a baby, something so small, become something so big? Like, how does that happen?
Did you know that little pieces of you are dividing into two every day?
Pieces of me don't divide every day. That'd be weird.
Dude, check this out.
What is happening to me right now?
Those little things that divide are actually cells. Mitosis is a part of the process known as the cell cycle. The cell cycle regulates cell growth and replication, or mitosis. The growth of the cell occurs during interphase, which includes three parts: G1 phase, S phase, and G2 phase. The G1 phase makes the decision to divide the cell. S phase replicates DNA and condenses chromosomes into sister chromatids joined at the centromere, and G2 phase prepares the cell for division.
The next phase is M phase, or mitosis. Mitosis involves 4 steps: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin condenses into chromosomes, the nuclear membrane breaks down, and the centromeres migrate to opposite sides of the cell. In metaphase, the chromosomes are lined up in a straight line, a position called the metaphase plate. The centromeres of the chromosomes become attached to spindle microtubules. In anaphase, centromeres of each chromosome separate. Each sister chromatid becomes its own chromosome as spindle microtubules cause the homologous chromosomes to move toward opposite poles of the cell. In telophase, the chromosomes gather at poles and begin to form daughter nuclei. The nuclear membrane begins to form around these chromosomes, and the chromosomes unwind, becoming chromatin. Simultaneously, cytokinesis occurs. Cytokinesis is when the cleavage furrow develops in the middle of the cell, and the cellular membrane breaks, and the parent cell becomes 2 identical daughter cells. Each of the daughter cells will have the same number of chromosomes as the original parent cell. Okay, Eric, and now I understand mitosis. So, what's meiosis? Why is it different?
Meiosis is the process by which haploid nuclei replicate during sexual reproduction. It is used to produce haploid daughter cells. This is achieved through 2 rounds of chromosome separation, known as meiosis I and meiosis II. This causes the genetic material to replicate once before meiosis, and divide twice during meiosis, giving all 4 of the daughter cells half the number of chromosomes as the original parent cell. Meiosis I separates homologous chromosomes. It involves 4 stages: prophase I, metaphase I, anaphase I, and telophase I. These stages behave the same way in meiosis I as they do in mitosis. They even have the same outcome: 2 daughter cells with the same number of chromosomes as the original parent cell. In prophase I, chromatin condense into sister chromatids. At this point, tetrads can cross over on sister chromatids, causing mutations in the genes. During metaphase I, the sister chromatids line up and become attached to a spindle microtubule. These spindle microtubules pull sister chromatids apart during anaphase I. Finally, in telophase I, 2 separate cells form, with an equal number of sister chromatids in each cell.
Meiosis II is when sister chromatids separate. The 2 cells with the same number of chromosomes will divide into 4 new cells with a slight variation in genetic material and half the chromosomes as the original parent cell, and the 2 cells dividing. The 4 steps are similar to meiosis I and mitosis. Prophase duplicates connecticores. In metaphase, the chromosomes line up. In anaphase, the sister chromatids separate. And then, through telophase and cytokinesis, we are left with 4 haploid daughter cells. Each will have half the genetic material as the parent cell. Okay, Eric. Now I understand how cells divide. In meiosis, they end up with half the chromosomes so that all the cells don't end up the same. And then this way, when 2 haploid cells come together, they can create a brand new cell with new genetic material inherited from both parent cells.